Hello friends, welcome to another video tutorial, and today we will discuss about the mitochondria. First of all, m- mitochondria was seen by Colliker in 1850 in the muscles of insect, in the cells of muscles of insect, and he named them as cytoplasmic granules. Later on, in 1894, Altman called them as bioplast, and further Benda in 1897 named them as mitochondria, and the structure is given by Palade. Similarly, mitochondria inner membrane contains some particles which are known as F particles discovered by Fernandez Moron in 1961. In 1967, Racker says that the enzymes for Krebs cycle are present in mitochondria. And later on in 1963, Nas and Margit shows that the mitochondria also contain a DNA, and the length of DNA is 5 micrometer, and this DNA is GC rich. That's why mitochondria is known as autonomous cell organelle, and it is not completely autonomous because it only produces its 40% protein, and for its 60% proteins it depends upon cell DNA, or nuclear DNA. That's why it is known as semi autonomous cell organelle. Let us discuss about its structure and function. First of all, we will discuss about its structure. As we know, the structure is proposed by Palade, and if we go through the structure of mitochondria, we found that mitochondria is 3 to 4 micrometer in length and zero point 0.5 to 2 micrometer in width. Okay, it's look like a kidney or bean shape. Okay, if we draw a diagram of mitochondria, we found that the have two mitochondria have two membranes have two membranes. Outer membrane is smooth, and a inner membrane which shows so many folding inside like this. And this inner membrane is known as Christie, and it is an smooth outer membrane. Christie is inner membrane. Okay. The space between two membranes is known as perimitochondrial space. Okay. And this perimitochondrial space is filled with H plus ions. It contains large amount of H plus ions. Okay. Inside this crusty, it contains toward inner side some dot-like structures which are known as F1 particles. Yeah, F particles who have three parts. One is F0 or F0, which embedded inside the membrane, and this type and this round structure present on it is known as F1 particle. It was discovered by Fernandez and called, that's why they are known as the F particle after his name. It contains its own DNA, which is known as naked, or it is just like prokaryotic DNA. That's why it is known as circular DNA, which is GC rich. GC content is high and it produces 40% of protein or enzyme required by the mitochondria. That's why it is semi-autonomous. The liquid filled inside the mitochondria is known as matrix, known as matrix, and it also contain some ribosomes. It also contains some ribosome, or sometime we also see polyribosome. And these ribosomes are 70S type of ribosome, 70S type of ribosome. Okay. Apart from this, if we deeply study the inner membrane of mitochondria, we found that it contains one is these F1 particle and four other proteins on this surface. Four other proteins in the surface which are known as transporters, yeah, electron transport chain, ETC, ETC, electron transport chain protein transporter, and they are known as the 1, 2, 3, and 4. Okay. Next thing is very important that the mitochondria, uh, origin of mitochondria. By looking at structure, we found that the mitochondria contain a circular naked DNA, which is just similar to prokaryotes. Okay, first point. Second point is that uh, if we see that the origin of another mitochondria, so mitochondria divide by binary fission, just like bacteria, and produce another mitochondria from previously exist mitochondria. That's why it is also shows the character like prokaryotes. Second point. Okay. And it is think that once upon a time this mitochondria may be a bacteria which was engulfed by the eukaryotic cell and which provide it shelter and food. In return, it get Krebs cycle or complete digestion of glucose from mitochondria. So, such type of views shows that mitochondria is present in symbiotic association with cell or it is inside the cell, so it shows that it is an endosymbionts. Endo यानी कि inside and symbionts means symbiotic association between two living organisms in which both are present in mutual cooperation and benefit to each other, or in which one is known as symbionts partner. So it is an symbionts to eukaryotic cell. Okay. So it is think that this is the theory. I already told about it is known as semi-autonomous cell organelle because it can produce its own protein of 40% and 60% depend upon this. Now, let us discuss about the functions of mitochondria. If we see that the number of mitochondria inside a cell increase if cell is metabolically active. If a cell is metabolically very active, the mitochondria number is increases in such cell. For example, an average cell contain 1000 to 1600 of mitochondria. But if we go through liver cell, their number may be high, up to 5000. Another example, a amoeba, which is known as chow chow, cons- contains approx 5 lakh of mitochondria. 5 lakh of mitochondria. Similarly, a algae, which is known as macrostis, contain only one mitochondria per cell. So, the muscle cell of a worker or which do work of a labor, hard work, lifting the weight, have more mitochondria in comparison to a person who did not do such things. So, the mitochondria number may be increases or decreases according to their needs. And average lifespan of a mitochondria is 5 to 10 days. For this time period, they live inside a cell and get die and another mitochondria is produced. Okay. So this is their number. So mitochondria number increase or decrease according to requirement of cell. They are mostly present in a highly metabolic cells in which regularly energy requirement is needed. Okay. In mitochondria, a process is running on which produce ATP by the breakdown of glucose that is known as oxidative phosphorylation. Oxidative phosphorylation. This glucose in cytoplasm break down into pyruvic acid, and this pyruvic acid is further converted into acetyl coenzyme A, and this acetyl coenzyme A leach into mitochondria, and where it again converted into pyruvic acid and combined with oxaloacetic acid to produce citric acid. That's why this cycle is also known as citric acid cycle, tricarboxylic cycle, or TCA, or discovered by Hans Krebs. That's why it is also known as Krebs cycle, where the citric acid is further converted into succinic acid, alpha-ketogluteric acid, malate, fumarate, and oxaloacetic acid. In during this process, it produces NADPH2 and FADH2. This NADPH2 is converted into ATP, and this FADH2 also converted into ATP, and this process is known as phosphorylation. This NADPH is breakdown by NAD dehydrogenase enzyme, and electrons are get free from the oxidation of this, and the final electron acceptor is oxygen, and these electrons are transferred from one acceptor to another carrier or acceptor and finally reach to the oxygen, है ना? And we will study its how it work in details in the respiration chapter. However, I will do give some mm, here also. As you seeing there, this is the NADH molecule which is produced during the Krebs cycle. In the presence of NADH dehydrogenase enzyme, it releases its electron which are accepted by flavin mononucleotide, and this flavin mononucleotide transfer this electron to FES, and this is finally accepted by ubiquinone. This is finally accepted by ubiquinone. Ubiquinone converted into ubiquinol. It is a partial site. It is interconvertible ubiquinone to ubiquinol, and ubiquinol it gets circulated in between these two till that the cytochrome B accept these electron. Okay, as the cytochrome B accept these electron, then it transfer it to FES, and finally these electrons are accepted by cytochrome C. Okay, cytochrome C, and later on this cytochrome Ca send this to send these electron to the copper, and from where they are transferred to cytochrome A, and finally they are transferred to the oxygen molecule, from which after getting this water is produced, which is known as new water. So here we saw that in NADPH, I already told you there is four complex, one, two, three, four, and last one is five. Five is this, which is known as ATP synthetase. ATP synthetase pump or F1 particle, which are discovered by Fernandez. So this and before this, they four are included. So during NADH, only one a complex one accept the electron and transfer to 3, 3 to 4, and the hydrogen ions are transferred to the perimitochondrial space, like there. Okay, and from there they are come inside via this F1 particle. And during this, an ADP is for, I will told it a bit right now. And now, if we go through the second channel, in which a FAD molecule, in which a FAD H2 molecule is break down with the help of enzyme succinate dehydrogenase. Succinate dehydrogenase. And electron are released, which are accepted by 3, 4, and finally 5 to create an ATP. So, alternatively, 1 and 2 are utilized. One complex is utilized in NADS breakdown, and complex 2 is utilized in FADH case. Okay. Now, as the electrons are transferred and during this hydrogen ions are released inside the this lumen here, H plus ion release. Now these H plus ions migrate inside. These H plus ions are migrated inside by the use of this F1 particle. What happened here, as these H plus ions are come, this is the F0 node and this is F1, and this is its stock. Okay, so what is this in here? In this F1 particle, there is three sites. There is three sites. One site is for ATP and ADP and inorganic phosphate binding site. Second site is for ATP, yani this, and third site is free. So there is three sites, and the energy released during the movement of this H plus ion inside the cell from outer space or perimitochondrial space is consumed to produce a bond between this ADP molecule and inorganic phosphate to produce ATP. As these hydrogen ions come inside and a electrochemical electro potential difference is produced due to presence of H plus ions in outer space here and the charge inside this matrix. So electropotential complex is produced due to which this particles are rotating. It start rotating in this direction, and this rotation produce an energy actually, which produce ADP and inorganic phosphate bond to produce ATP. So This F1 particle contains an enzyme which produce bond in between these two molecules. That's why this is known as ATP synthetase pump. Okay, and the ATP is produced. I think so. You get to know. So the F mitochondria number is high in highly metabolic. For example, the middle piece of sperm contain very high amount of mitochondria to provide energy to it for movement. When sperm swing inside the vagina to reach the ova, it required energy. So sperm contain high amount of mitochondria. Similarly, during vitellogenesis, formation of egg, the number of mitochondria is very high in these cells. Thank you so much. I hope you will get understand something. If there is any problem, kindly let me know. And thank you so much for your concern and hope so you will get understand it. Thank you.